Welcome back. Today we'll be going through the small intestines. So, the principal site of digestion and absorption of degraded food is the small intestine, and it runs from the pylorus to the ileocecal valve. And the walls are kind of the same throughout the alimentary canal, except for this one has serosa instead of adventitia. And the small intestines are characterized by the plicae, and the plicae circularis is when you look on it like laterally, it looks like these like shelves, but they're actually spirals, and they're lined by a dense covering. Like, they're so dense you can hardly like pick them apart of the intestinal villi, and those will project into the lumen of the small intestines in order to increase the surface area for maximum absorption of water or nutrients. So here are the villi. This is going to be the lumen, the mucosa, and the smooth muscle. So the mucosa is going to be made out of a simple columnar epithelium, and it's going to lie on top of the lamina propria, which has a lot of blood and lymph capillaries and a few strands of smooth muscle. The plasma cells are going to be the immunoglobulin A producers, and they're also eosinophils and macrophages. The lymphocytes will form the lymphoid nodules, and they aggregate in the ileum mostly. The muscularis mucosa is a smooth muscle, and they all together extend into the intestinal villi and form the core. And the core contains all of these, and lacteals are just ducts. The submucosa is going to be innervated by the Meissner plexus, and then the duodenum will have the Brunner's glands, and that makes the alkaline mucus in order to break down the triglycerides in the food and to protect the small intestine from gastric secretion. And they extend into and form the core of the plica circularis together with the mucosa. And then the muscularis propria is going to have a couple layers again. The Auerbach's plexus is going to innervate the submucosa and provide the peristaltic movement. Remember, while the Meissner will kind of control the constriction aspect. And then the mesentery will be attached to the serosa to everywhere except for at the duodenum. There's going to be a bare area there. Plica circularis, as you can see here, is mostly only in the jejunum, and they're also known as the valves of Kirkring, and they're made out of both mucosa and submucosa. So in the first few centimeters of the duodenum, it's going to be absent along with the distal part of the ileum, so which is like at the very beginning and the very end. It's the best seen in the jejunum, and they are going to have the villi on top of it. And between the villi, so in between them, is going to be the crypts of Lieberkühn, which once again secretes some kind of alkaline solution. Okay, so the villi are going to be mucosa overgrowths, and they are covered by a simple columnar epithelium, along with the absorption cells and the goblet cells. The core will have the blood vessels that transport the amino acids and other things to the hepatic portal circulation. And the lymph, or the ducts, are going to run to the submucosa, and those transmit chylomicrons, not just lymph, to the thoracic duct, to the general circulation, and that bypasses the liver, because this has already been processed. Why do you need to process it again, So the muscularis mucosae is going to enhance the drainage due to peristaltic movement, and then each absorptive cell of the epithelium will form lots and lots of microvilli. These are going to be specialization of the apical surface of the enterocytes, held together by microfilaments, and they integrate into the cytoskeleton of the cell body, and they make up the brush border. Each of the villi are going to be coated by a glycocalyx, and that provides both protection against the autodigestion due to the acidic environment and a site for the absorption of pancreatic enzymes. So the enzymes will like dock and then be able to like reabsorb and reduce, reuse, recycle. So the creeps of Lubricune are the simple tubular glands. As you can see here, they're tubular, they just kind of jut upwards. And they're formed by invagination of epithelium. And the lamina propria grows down to meet the muscularis mucosa. And those will secrete intestinal juice, which is rapidly reabsorbed. And it contains enteropeptidase, which will convert the trypsinogen to trypsin. So you can see here, here's the crypts. So they'll just empty directly into the lumen. Enterocytes are going to be the absorption cells, and they are going to be quite tall and columnar. And then the lateral cell membranes are going to be joined by desmosomes and tight junctions. And those seal off the gut lumen, and they are a site of sodium and potassium ATPase activity to regulate, of course, the reabsorption of salt. In the cytoplasm, it's going to have plenty of lysosomes, smooth endoplasmic reticulum, rough endoplasmic reticulum, the mitochondria, and Golgi complex. It's a really blurry cell, but here you can see there's a brushed border, and right below is the terminal web. You can't really see the Golgi apparatus, but here's the nucleus. Here's the basal cytoplasm. And they only survive about like 5 to 6 days. Besides absorption, the microvilli will also secrete disaccharides and peptidases, and those bind to the microvilli and hydrolyze those respective surfaces or substances into monosaccharides and amino acids. The digestion of fat is due to the pancreatic lipase, along with bile from the gallbladder, and most of the liquid absorption will take place in the duodenum and the upper duodenum. So goblet cells are unicellular mucus-secreting cells. They kind of like lie between the absorption cells, and they get more numerous when you're like going towards the ileum. The cytoplasm doesn't have actually like a lot of mucin, and the glycoprotein mucus is going to be hydrated and cross-linked to form the mucus, and those will be secreted by the goblet cells to protect and lubricate the lung, along with like moving food down. So here are the goblet cells. They're like pretty superficial. Paneth cells are going to be exocrine cells, and they have a cytoplasmic, they have a basophilic cytoplasm, along with a basal rough endoplasmic reticulum. They have secretory vesicles that contain defensins, some kind of immunological substances that help defend the body, the lysosomal enzymes and the zymogen granules. The zymogen granules can be broken down and actually break down the membranes of microorganisms and bacterial walls. Not necessarily a good thing, because we do need our gut flora to convert a lot of larger, more complex carbohydrates into simpler carbohydrates, which we can use. They have some role in innate immunity and regulating the microenvironment, but it's still kind of mysterious. They last for weeks. So enteroendocrine cells are going to be found in the lower third of the crypts. They're going to be present all along the smooth intestines, and they secrete plenty of peptides, and they represent part of neuroendocrine system. When they're stimulated, they will release their secretory granules along with the hormones, and they control peristalsis and regulate secretion for food digestion and a sense of satiality. Here are the enteroendocrine cells. they lie kind of far away from the lumen. Crypt cells, on the other hand, are fairly close. Okay, so the stem cells lie at the bottom of the glands, and they kind of push up to replace whatever. And the intermediates, well, they're still like, switching and differentiating. They have the features of both the enterocytes and the goblet cells. So there's both absorption and secretory function. And those will replace the enterocytes and the other goblet cells every 4 days. So the turnover in the gut is pretty fast. The duodenum has the Brunner's glands, and those will secrete, once again, the alkaline mucus. And the duodenum, under like histocides, they're going to be much less frequent goblet cells, hardly ever any plica circularis, and few lymphoid tissue. The jejunum will have the most plica circularis. They will also have the longer villi, and they don't have any Brunner's glands or the Peyer's patches. The ileum will have the most lymphoid tissue that shows up as the Peyer's patches, and the epithelium will have cuboidal and M cells, and they also have much less goblet cells and villi and plica circularis, so it wouldn't be there at all. M cells are microfold cells, and those are modified epithelial cells. And those are found between the columnar epithelial cells that cover the Peyer's patches. So here's the lymph follicle, that's in like the base near the germinal center, and then the Peyer's patches, like the entire unit. The intercellular pockets open to the underlying lymphoid tissue, and the basement membrane is pretty porous, allowing things to go in and out easily. As you can see here, tight junction will keep the cells together, but they have to bind, and then the body's going to be like, phagocytosing it. So the antigens in the gut lumen are going to be bound by the M cells, and they go through transcytosis into their intraepithelial pockets, where the dendritic cells will take up the antigens, process it, and present it to the T helper cells. Only the T helpers and the dendritic cells leave the M cells, and they leave through the poles of the basement membrane. Here, B cells, T cells, dendritic cells are allowed to leave. These guys kind of have to stay behind. They interact and initiate the immune response to the antigens and form the secondary lymph nodules. B cells are going to be stimulated by T cells, and those differentiate into plasma cells, and those secrete more IgA. So as you can see, lymphocytes goes, dendritic cells relay information, M cells are right here. So injury can occur because of something called celiac sprue, or gluten intolerance, or celiac disease. And there's just going to be a whole lot of information, blunting of the villi. Problem is they're going to basically clean out the villi. Everything's going to be too smooth, and there's going to be malabsorption of food and nutrients. There's not really any cure. There's nothing to take either. Not really any surgeries. In order to really manage this, you have to have a strict diet. So the cholera toxin will kill the epithelial cells by disrupting the cell junctions, allowing fluid to come in, and there's going to be a loss of the integrity of the fluid barrier between the lumen and the lamina propria. So the body fluid will move freely into the lumen of the intestines and lead to watery diarrhea. But it's not just a one-off kind of thing. This is rapid, massive dehydration. So you had to treat with some kind of IV plasma substitute or oral, salty water. Cholera vibrios clears in a few days, and then epithelium will restore itself if treated. If not, the person will die from hypovolemic shock. That's it for now. Thank you for your attention, and good luck studying.